Hi, my name is Rusha Patel. I'm going to be presenting chapter 15 for the unit project. So, the chapter 15 is of saccharides of physiological significance or carbohydrates. And I'm going to explain the types, structures, and biological roles of carbohydrates, also known as saccharides. And I'll also talk about the importance of energy metabolism, structural function, and disease. So, the carbohydrates are essential biomolecules that exist in all plants and animals. Because of their high polarity, they participate in a wide range of biological processes and dissolve readily in water. Their primary functions are metabolic and structural. In terms of structure, they are a component of plant cell walls and animal connective tissues. They give energy through metabolism. Glucose is the main fuel used by animals and even by fetus as a universal fuel. Additionally, carbohydrates serve a building block for bigger biomolecules such as lactose, glycolipids, galactosemia, glycogen storage diseases, fructose intolerance, and lactose intolerance are associated with the metabolism of the carbohydrates. What are carbohydrates? The primary constituents of carbohydrates, also known as saccharides, are carbon, hydrogen, and oxygen. Although not all saccharides meet the exact formula, the general formula um, for is frequently expressed as CH2O2, but not all saccharides strictly follow this ratio. Because certain sugars also contain additional elements such as sulfur or nitrogen, biochemists prefer this name saccharide. From a structural perspective, all saccharides are in polyhydric alcohols with ether or ketone or aldehyde group, which determine whether they are preferred to as ketosis or aldoses. Because they serve as important source of energy, are a component of nucleic acids and support structural framework in both plants and animals. Carbohydrates are essential to life. The versatility of simple sugars is demonstrated by the fact that they can be combined in over 1,000 different ways to create a wide variety of trisaccharides. Additionally, some carbs can be performed into sugar alcohols like xylitol or sorbitol, although they can result in intestinal gas when poorly absorbed. They are frequently used as sweeteners for diabetics since they offer sweetness with less calories. Though not all of them are sweet, carbohydrates are generally quite polar and soluble in water. This sweetness can be replicated with little to no calorie addition using artificial sweeteners. Carbohydrates are characterized according to on how many sugar units they contain. Glucose and fructose are examples of monosaccharides, which are single units that cannot be further broken down. Glycosidic linkages bind two monosaccharides to form disaccharides like lactose, maltose, and sucrose. 3 to 10 sugar units make up oligosaccharides, which are frequently indigestible by humans. Lastly, long chains of sugar make up polysaccharides like cellulose, glycogen, and starch, which are either structural elements in plants, animals, or molecules that store energy. Because they include numerous asymmetric carbon atoms, carbohydrates can create several isomers. Nearly all sugars that are biologically active are in the D form, and the D and L designation indicate the orientation of hydroxyl group on the last chain. last chiral carbon. Depending on the orientation of the hydroxyl group on the anomeric carbon, sugars can also be classified as alpha or beta anomers. Glucose and galactose are examples of epimers, which are sugar that differ only in one chiral carbon. Because fructose is a ketone sugar and glucose is an aldehyde sugar, they serve as example of aldose ketose isomerism. Lastly, when monosaccharides cyclize in solution, they can form either five-membered furanose rings or six-membered pyranose rings. There's two types of glucose, cyclic and straight chain. Because the structure is more stable, it primarily manifests in the cyclic or hemiacetal form in solution. In equilibrium, these are two primary cyclic forms, alpha D-glucopyranose and beta D-glucopyranose, with the beta form being marginally more prevalent. A chain conformation diagram, a half-forward projection, or a straight chain formula can be used to depict these structures. Knowing these structures make it easier to understand how glucose behaves through metabolism and form of bigger carbs. A glycosidic bond is created when a monosaccharide hydroxyl group on its anomeric carbon combines with another substance. The linkage is referred to as a O-glycosidic bond if it forms a bond with another hydroxyl group, and an N-glycosidic bond if it forms a bind with an amine. For monosaccharides to become disaccharides and polysaccharides, these linkages are essential. In biology, ribose binds to adenine in nucleotides like ATP, forming an glycosidic linkage. Glycosides are also significant in medicine. For instance, antimicrobial glycosides like streptomycin are employed in medicine, while cardiac glycosides like digitalized albumin influence heart function. Glycosidic linkages bind two monosaccharide molecules to form disaccharides. The three most well-known are lactose, maltose, sucrose. Cane and beet sugar contains sucrose, sometimes known as regular table sugar, which is composed of glucose and fructose. Glucose and galactose combine to form lactose, which is present in milk. Lactose intolerance is caused by lack of the enzyme lactase during the digestion of starch maltose, which is made up of two glucose molecules shown by an alpha 1 to 4 bonds occur in as an intermediary. This disaccharides show how various link provides unique digestion patterns and functional characteristics. Long chain of monosaccharides molecules make up polysaccharide. The primary energy storing molecule in plants is starch, which is composed of linear amylose and branching amylopectin. Similar storage functions are carried out by glycogen in animals, although it is much more densely branched. Enabling quick energy release, contrarily, cellulose, which is indigestible by humans and contributes to dietary fibers, include beta 1 to 2 connection, 1 to 4 connection, and is the structural element of plant cell walls. Inulin, a fructose polymer, is another polysaccharide that is utilized in clinical studies to gauge renal filtration efficiency. We have complex carbohydrates that serve certain biological purposes in addition to simple sugars. Cartilage and connective tissue contain amino sugars and such glucosamine and galactosamine. Hyaluronic acid Chondroitin sulfate and heparin are examples of glycosaminoglycans that assist lubricate and cushion joints and have anticoagulant properties. The hydrated matrix that support connective tissue is made of protein called proteoglycans that are connected to lengthy sugar chains. Cell membranes and plasma lipoprotein contain glycoproteins and glycolipids, which are essential for cell recognition and signaling because they make sugars with proteins or lipids. About 5% of cell membranes bulk is composed of carbohydrates. The glycocalyx, a protective and communicative layer on the cell surface, is made up of glycoproteins and glycolipids. The ABO blood group system is an intriguing illustration of the diversity of carbohydrates. Certain oligosaccharides chains affixed to red blood cells protein identify each blood type. And acetylgalactosamine is added by type A cells, galactose is added by type B cells, and neither addition is present in type O cells. These minute variations in sugar levels are sufficient to cause immunological reactions in blood types that are incompatible. The rate at which a carbohydrate elevates blood sugar levels after eating is measured by the glycemic index, or GI. Low GI foods like oats and lentils provide a slower, more regulated release of glucose in the bloodstream, whereas high GI foods such as white bread or sugary drinks induce rapid increases. Since it enables people to select carbs that maintain steady energy levels and avoid excessive insulin responses, knowledge of the glycemic index is crucial for treating diabetes and fostering metabolic health. Here are my five multiple choice questions. Which sugar is a ketose? The options are glucose, galactose, fructose, or ribose. Number second, the D and the L designation refers to option A, optical rotation, option B, position of carbonyl group, option C, configuration around last chiral carbon, or option D, type of glycosidic bond. The third question is lactose is composed of glucose plus glucose, glucose plus galactose, glucose plus fructose, fructose plus galactose. Option 4. What is the structural carbohydrate in plant cell walls? Starch, glycogen, cellulose, inulin. Number 5. Glycoproteins are found in cytoplasm, DNA, cell membranes, or mitochondria. Here are my citations. Thank you.